Respected examiners, my supervisor, and all present here. I am Navpreet Kaur, a research scholar at School of Computer Science and Engineering, Lovely Professional University. It's an honor to present the final viva of my PhD thesis entitled A Novel Soft Computing Based Approach for the Detection of Leukemia Disease. First and foremost, I would like to express my sincere gratitude to my PhD supervisor, Professor Dr. Amar Singh, for his support, guidance, and encouragement throughout this research journey. This research primarily focuses on the development of the soft computing based approaches for the efficient and the accurate detection of leukemia. Leukemia is a type of the blood cancer which occurs when the blood cells are developed in the large number inside the bone marrow. So with the brief introduction to the title of the thesis,
स्लाइड आपने चेंज की है? अभी नहीं किया ना? हाँ ठीक, नेक्स्ट करना, देख लें, मतलब चल रही है ना? हाँ ठीक है, चलिए.
So with the brief introduction to the title of the PhD thesis, now I would like to walk you through the presentation structure which I will be discussing today. So firstly, I'll talk about the topic. I'll be introducing the background of the research.
अभी स्लाइड पे आ सकते हैं, ठीक है, ये पता लग गया कि this is your sequence, so showing this is good enough.
Okay, so now if I talk about the introduction to the things, introduction to the topic, first of all, it's about the blood. So blood is considered both a tissue as well as a fluid, and generally the purpose of the blood is what? It contains various types of cells. 4 different types of cells are there: white blood cells, red blood cells, platelets, and plasma. So red blood cells are also called as erythrocytes, and white blood cells also known as the leukocytes. Then we have the thrombocytes.
आप अपने काम की तरफ बढ़ो, introduction.
Okay, now then the blood cancer.
हाँ.
So how actually blood cancer occurs? We are having one bone marrow. Bone marrow is what? It is a spongy tissue which is inside various bones. One of that bone is like the pelvic bone, the skull bone, the spinal bone. So over there we are having the bone marrows. Bone marrow generate the stem cells.
आगे आ जाओ, ठीक है, अपने काम की तरफ बढ़िए, ठीक है.
So these are the types of blood cancer.
Fine.
These are the traditional ways of detection of leukemia.
Right.
Different tests and the issues with the traditional methods.
Traditional methods, medical methods, ना, you mean?
Yes, sir. These are the
उनके
medical.
साथ issues हैं. Now you come to your domain.
So soft computing and hard computing approaches. Then coming to literature review.
Yeah.
So here, this is the methodology for conducting of literature review. Initially started with the 650 papers from the various reputed journals and all, then filtered out, and ultimately I have focused on the 145 articles. So this is the summary of the various machine learning based models for leukemia classification.
इधर राइट में क्या स्लाइड नंबर लिखा हुआ है या?
So here?
कहीं स्लाइड नंबर है स्लाइड पे?
नहीं sir.
मैं कैसे नोट करूँ?
Sir, it is 14.
14, ठीक है, तो कोई बात नहीं, आगे जब भी हो ऑलवेज कीप स्लाइड नंबर ऑन द स्लाइड सो दैट दिस इज विजिबल.
Okay, sir, sure.
हालांकि अभी भी हो जाएगा, पीपीटी में डालोगे, बट वो है, अब मैं बीच में ही पूछ लूँगा, अदरवाइज नॉर्मली आई नोट डाउन एंड देन लेटर आस्क. तो उससे फ्लो नहीं टूटता आपका.
So these are
जैसे
the
पिछली स्लाइड पे आओ बिफोर यू गो, अदरवाइज अब देखिए इसमें आपका फ्लो चार्ट का फोल्ड कितना छोटा रह गया, राइट में वेस्ट स्पेस हो गया. ये ऊपर आप एक बार सिर्फ हेडिंग में लिख देते कि लिटरेचर का मेथड है, ये सारे स्पेस को जगह मिलती. बॉक्सेस के अंदर फॉन्ट बड़ा होता तो बॉक्स दिखाना पर्पज नहीं है, बॉक्स के अंदर क्या लिखा है वो पढ़ना पर्पज है, वो मैं नहीं पढ़ पा रहा हूँ. और डिस्टेंस के साथ कोई और होगा तो अगर यूट्यूब पे जाएगा तो बिल्कुल ही नहीं पढ़ा जाएगा, कितना छोटा रह गया. तो स्लाइड में कंटेंट्स आर इम्पोर्टेंट. उधर आपने जो एरो से लिखा है वो भी नहीं पढ़े जा रहे. बॉक्स का 30% स्पेस यूज्ड है, 70% खाली पड़ा है. बड़ा फोन तो उसी स्पेस में सब कुछ बड़े से पढ़ा जाएगा और स्पेस भी यूज हो सकता है. अगेन अब इसमें देखो, जब आपने कलर का कंट्रास्ट लिया, देखो आपके जो लेफ्ट मोस्ट स्टडी के कॉलम है वो कंट्रास्ट में छुप गया. बहुत जोर लग रहा है पढ़ने में, और गर्ल्स को तो कंट्रास्ट की हमसे ज़्यादा अकल होती है, नहीं? हाँ, कलर्स की भी वही कह रहा हूँ. तो वो ये सारी चीज़ें ध्यान देनी है. ठीक है, आगे चलो. हाँ बता.
So these are the another machine learning based models for the leukemia classification, and here it's mentioned what data sets are used in the various studies, the methodology they have used over there, accuracy and the limitation. So mostly the data set which have been used in the studies is either ALL, IDB or certain images, very small number of images are used over there, and very rarely the large data set is used. Now next to this is the deep learning based approaches for the leukemia detection. Here different data sets are used in this. The methodologies which are used are also mentioned.
हार्ड बाउंड लाए हो आप थीसिस दे दो मुझे, मतलब साथ में इसी से ही देखता हूँ। ठीक है, ये रिवाइज्ड वाली है ना?
हाँ जी।
ठीक है। So you can come towards research gaps quickly, ठीक है? ये भी आपको क्योंकि आपकी रिपोर्ट इवैल्यूएट कर ही चुका हूँ, तो I have gone through all these things here. Yeah.
Now these are the research gaps. So what is mainly analyzed from the existing literature and the studies? First thing is the insufficient data set. The size of the data set is very small and diverse data sets are used, like some images are taken from the hospitals only 100 images or sometimes only 50 images. So this is one problem here. Another is the deep learning models, they give the good results, but the size of the models are very large. There are very large number of layers. Third thing is,
what are your objectives? ठीक है, हाँ बताइए.
Objectives here is the first objective was to study, analyze and evaluate the performance of various existing soft computing based approaches for the detection of leukemia disease. Second is to propose a novel soft computing based approach for detection of leukemia disease, and third is to compare the proposed approach with the existing approaches using few performance metrics. So these are the three objectives.
Okay.
To accomplish these three objectives, the major contribution of this thesis, firstly, I have proposed one approach, VGG16, PCA and PB3C. Secondly, proposed another approach, VGG19, PCA and VBBC. And third is a hybrid parallel 3-parent genetic algorithm. And
what is difference between VGG16 and 19?
So it comes in terms of the layers. Like in case of VGG16, we are having the 16 convolution layers, whereas in case of 13 convolution layers, whereas in case of VGG19 we have total 16 convolution layers. So when the convolution layers are more, they fetch the more intrinsic features, more deep and depth features.
So why we should not have used VGG19 in the first point itself?
Because it takes more training time also, although it is fetching more features, but it takes more time and complexities also.
Training time is more, is that a big issue? Even if it takes more training?
So there is it is like a computational complexities there, it is taking more resources as well, more time also. So I compared these two. I have initially analyzed the results with 16 as well as 19 both.
Okay.
Then VGG16, PCA, PB3C, what actually the work of this one is published in the International Journal of Information Technology, Springer Journal. So what is actually in this approach, three things are integrated together. Here VGG16 is used to extract the features from the images, and then it is extracting 25,088 features from per image, and as these are the very large number of features, so we need to reduce it. To reduce that, dimensionality reduction PCA is used, which is reducing 25,088 features to 1145 principal components. And further, these are the components which have been extracted, and we need to select the most optimal one out of this for this PB3C parallel big bang.
Did you store the features after extraction, or did you implicitly supply it to the model being used?
It was supplied to the model.
Supplied.
Yes.
So did you evaluate or did you try to see कि जो models features generate हो रहे हैं उनको एक बार देखें, क्योंकि उसके बाद आप उस पर PCA लगा रहे हो, तो is there a necessity of PCA? You applied PCA one angle.
Yes, sir.
Was there a necessity of PCA to be applied?
Sir, if we were not applying the PCA, then it was accuracy was very much less.
Okay, accuracy based is what we say कि hit and trial हो गया. बट पीसीए क्यों लगाना चाहिए? एनी आइडिया?
sir,
अगर मान लीजिए कोई डाटा दिया हो तो आप कैसे डिसाइड करेंगे कि शुड आई अप्लाई पीसीए?
So there were redundant features also.
हाउ डू वी नो?
Because the same type of feature, same edges. If
बट यू डिडंट स्टोर द
Sir,
फीचर्स.
then we had created one matrix, like matrix file of all those features, where it was storing the values of the features. So redundant values were coming over there.
Redundant values in row or in columns?
So redundant value in terms of like row is for one particular feature, one particular image.
Column is for one
It is
feature.
in columns.
So columns were duplicated, you mean to say?
Yes, sir. So features were duplicate.
How many total features were there into that file?
Total features are
Total
like
columns, I mean.
Sir, it was our images of 450 by 450. So those number of pixels and same number of features were there.
अगर वो हर पिक्सल एक फीचर है तो पिक्सल्स में
So it was very
डुप्लीकेसी,
much. It
तो
was in
डुप्लीकेसी
thousands.
नहीं आए तो इट इज ऑब्वियस adjacent pixels will be always of the similar color until unless there is an edge. तो पिक्सल्स का कलर सिमिलर आना को तो हम डुप्लीकेसी या रिडंडेंसी नहीं कह सकते, किसी भी इमेज में आएगा वो तो.
But sir, it was like in our case of cell image.
अब सामने बोर्ड लगाया, मैं इसकी पिक्चर लूंगा, जब तक बोर्ड की बाउंड्रीज नहीं आएंगी तो पिक्सल सेम आएंगे ही. तो डज दैट मीन दैट आई एम गोइंग टू ट्रीट इट एज अ रिडंडेंट फीचर?
No, sir.
तो फिर कैसे, तो फिर तो
Sir,
आपकी
it was
स्टेटमेंट
like-
जस्टिफाई नहीं
It
हुई.
was like suppose this is one cell,
राइट
this is the another cell.
राइट
It's another cell. Now here the value, the color is different.
राइट
Edges over here.
Right.
But if I talk about the another image, in this case the value is different over here. Ultimately the values are in between 0 to 255
Right.
over here. So at this place there will be some less value, whereas if I compare the same position in this image, the value would be different.
ऑब्वियसली. लेकिन अगर आप एक इमेज में कह रहे हैं तो एक इमेज में उस पॉइंट से लेके अगले एज तक सारे पॉइंट्स की पिक्सल वैल्यू सेम है.
But sir,
इधर भी लगभग सेम एरिया में बहुत सारी सेम हो सकती है.
Yes, sir.
तो वो दोनों में डुप्लीकेसी वैल्यूज डुप्लीकेट होना आएंगी. शुड दैट बी ट्रीटेड एज अ डुप्लीकेट फीचर?
yes sir, they will be considered as the, like if this is a curve, but here it's not the curve, so these are the different
Okay.
features.
So if I say now I should just do the edge detection onto the image and after that I do my machine learning, then everything will be lost. I will just focus on the edges. मेरा सारा डाटा भी रिड्यूस हो गया, मेरी सारी रिडंडेंसी भी खत्म हो गई, मुझे पीसीए भी नहीं चाहिए, मुझे मशीन लर्निंग का भी मिनिमम रोल हो गया. Detect the edges and go ahead. That's it.
Sir, other than edges, the color, the intensity, the shape, everything was considered.
तो अगर वो है तो फिर वो रिडंडेंसी नहीं रही. If shape
हाँ जी. Sir, actually initially to extract the features out of this image, VGG16 was applied.
Okay.
VGG16 has extracted all the features from this image, and those
in
features
what
were...
form? In not in pixel form ना?
Yes sir.
या एग्जैक्टली अगर जो डायमेंशन पिक्सल्स की थी वही उतने फीचर्स बने.
Sir, the number of columns were those, they were in thousands.
राइट. तो
When
उतने ही
Yes, sir.
VGG16 के बाद आपके
Yes,
फीचर्स
sir.
बन गए.
Yes, sir.
ठीक है,
So-
आगे.
So there were like thousands of columns.
Okay.
Now, if we train the model based on all those 1,000 columns, it would be very complex and take a lot of time.
Okay.
To reduce it, what is done? Then PCA was used,
Okay.
so that the main features out of this is fetched, because this
तो
part
मेन
is
कैसे
unnecessary.
डिसाइड किया?
Like sir, cell area. Suppose here it is all the values are less.
पीसीए के बेस पे मेन आपने
So
डिसाइड किया
PCA
होगा.
is, in case of PCA, firstly the covariance matrix is calculated. So after that the eigenvectors, eigenvalues, eigenvectors, those things are calculated. It is doing what? It is comparing the covariance matrix is
ठीक है.
calculated.
उसके आगे
So then it looks the variance. We have
ठीक है.
considered the 99% variance over here.
99.
Yes sir.
ठीक.
So with the 99% variance, whatever features were there, we have considered those features. PCA has considered the 99% variance in the features.
99.
Yes, sir. Then out of this, it has extracted 1145 principal components.
Out of
Out of like 25088, this
25088. 1140.
Five principal components were analyzed.
ठीक।
Now further it has just
तो मैंने यही तो एक क्वेश्चन पूछा भी था, इसका आपने रिप्लाई में गोल मोड क्यों किया?
sir
कितने % रिडक्शन हुई है, मैंने यही पूछा।
sir, it was mentioned there like 99% variance is there and number of
1145 divided by 25008 is the reduction.
okay dimensional
है ना?
reduction.
तो कितनी रिडक्शन है, कितने % रिडक्शन है? आउट ऑफ 25,000 1145 रिमेन, तो 25088 minus 1145 divided by 25088, that becomes your percentage of reduction. समझ गई?
Yes sir.
चलो।
Okay, so next to this was the parallel Big Bang Big Crunch algorithm for the optimization of this, and it has selected 200 optimal features. Now this parallel Big Bang Big Crunch, so ultimately the number of features have been reduced over here. So this was the first approach, and how is it shown? Initially the data set is taken, provided to VGG16, then it has extracted the features reduced, PV3C for optimization is used here, then the fully connected layers for the classification are used, and ultimately it is classifying. Now VGG16, it's about VGG16, like it includes convolution layers, pooling layers, fully connected layers. In this we have the 13 convolution layers, 5 mix pooling, and
What is the role of textures here?
it's a role of
Or in your problem, textures, edges is okay, shapes is okay. Why textures should be bothered?
sir, like this is the image. So these are the textures. Somewhere it is whitish kind of thing, somewhere it is more dark purple, and it is telling whether the cell is the leukemic cell or not. So texture is important in that context. So these are the different layers as I told, and then it is already discussed, total 1145 principal components are selected. Now this is the algorithm, parallel Big Bang Big Crunch algorithm. Here we have done what? Initially in case of parallel Big Bang Big Crunch algorithm, firstly the population is generated, population of features is created over here. So we have created 4 population, n random populations can be created which are having the m candidate solutions. So candidate solutions are individuals and they are made up of the genes. So now in the Big Bang phase, the population is generally created, and in the Big Crunch phase, what we do is we just calculate the fitness of the Now here 200 principal components are selected, then fully connected layers are used for the final classification and the data set used.
Okay.
In this the CNMC 2019 data set is used. This data set is prepared at the laboratory oncology AIMS and it is containing 15,384 images. These are the pre-segmented images.
ठीक है.
These are the
फिर से colors and contrast देखो, है ना?
Sorry for
कोई बात नहीं, मतलब हाँ ये
Next time onward, she needs to work on that. Absolutely right. The visibility part. Yes.
Okay, so here the proposed approach VGG16 PCA PV3C is compared with the various existing machine learning based approaches and some of the studies from the literature review also, and it is analyzed like this model is performing well, it is outperforming all the rest approaches in terms of its accuracy, precision and all. So in the next one it's compared with the transfer learning approaches also VGG16. Simply firstly the model was trained with VGG16, detection was done with VGG16, 19, then PCA was integrated along with this, and after that the optimization algorithm was there. Now the next approach, that is VGG19, PCA and BBBC. Here in this approach instead of VGG16, VGG19 model is used, and VGG19 is having additional 3 more convolution layers which were not there in case of 16. The VGG19 layers also extract the 25,088 features. Here in terms of this PCA works little bit different. It is fetching 1222 features instead of.
Why PCA works different?
Sir, number of features again, but variance was as 99%. But in case of VGG 19, as it fetches the deep features, deep and detailed features due to its more number of layers.
अच्छा.
3 convolution layers are added as compared to 16.
How much it identity instead of
number of-
25088 पहले आपका वो था डाटा,
Yes,
इस
sir.
केस में कितना आया?
Sir, actually the number of features remains same.
Same.
Yes, sir.
But values were different.
Values were different. Because they are finding and extracting in detail and in depth. This is the only difference over here. So number of features remain same, and when out of these
But
20.
अगर आप कहती हैं कि वो नंबर ऑफ पिक्सल्स के हिसाब से है, that means there were 25088 पिक्सल्स.
Yes, sir.
तो फिर पिक्सल्स की इंफॉर्मेशन में तो पिक्सल का मतलब उस पिक्सल का आरजीबी वैल्यू,
But in detail, when it is looking
डिटेल
again and
का
again,
आरजीबी वैल्यू के अलावा और क्या डिटेल आ जाएगी
When
एक
it-
पिक्सल में?
when it looks for more textures. So
बट आरजीबी वैल्यू के अलावा भी कुछ पिक्सल की इंफॉर्मेशन होती है एक इंडिविजुअल पिक्सल की.
pixel is in terms of RGB only,
तो
like value is in 0
आप
to
कुछ भी
55.
कह दो वही आरजीबी वैल्यू ही तो आएगी.
Yes, sir.
तो फिर वो डिफरेंट कैसे आ जाएगी?
Due to the texture, color.
बट बेटा एक पिक्सल में तो ना टेक्सचर चेंज हुआ ना कलर चेंज हुआ, जो था वो तो वही रहेगा.
Yes, sir.
तो कैसे हो सकता है?
Sir, when it was provided, but the results were
तो
different.
you should know and say that probably should mean that what is statement you are making कि 25088 everything is just a pixel value, that statement becomes incomplete. अगर वो पिक्सल वैल्यू है तो दोनों में सेम ही रहेगी और उसके अंदर सिर्फ आरजीबी आएगा, कोई और चीज बची भी उसमें. एज आनी है तो वो एज तो एक पिक्सल से दूसरे पिक्सल की ट्रांसफॉर्मेशन में आएगी.
Yes, sir.
उस एज से तो कोई एक्स्ट्रा इंफॉर्मेशन पिक्सल पे तो मिलनी नहीं, उस पिक्सल का तो वही रहेगा. तो अगर आप वो कह रही हैं तो द फीचर्स एक्सट्रैक्टेड वर्सेस द स्टेटमेंट यू आर मेकिंग, देयर इज अ गैप. ठीक है, पढ़िएगा आगे उसको फीचर में, कोई बात नहीं.
Next to this was PCA was added, and after PCA, BBVC model was used over.
One of aspect of PCA is orthogonality evaluation. Are you aware what is orthogonality? तो जब PCA लगाया जाता है तो पता होना चाहिए कि why PCA and how it is related to orthogonality, and after PCA is orthogonality changing or we have done or not. उससे पहले मल्टीकोलिनियरिटी डिड इट एग्जिस्ट और नॉट, वाज इट इवैल्यूएटेड. तो वो लगाने के लिए आइडियली दैट इज अ प्री प्रोसेसिंग स्टेप टू चेक एंड ओनली देन पीसीएस वर्कनेस. और फिर 99% चाहिए या कितना चाहिए, व्हाट वाज द थंब रूल, हाउ व्हाई 99%, कोई रीजन था?
Sir, because when I analyzed, 95% variance was generally used, 95 to 99%.
Okay.
If we keep it 100%, then there is no any change. All the features would be taken as
Obviously,
it
100
is.
पे तो नहीं आएगा.
So, in case of medical terms, when we deal with the medical data, over there 99% variance was being
मैं कह रहा
considered.
हूँ, why not 98%? Why not
So
97?
we could consider that also, but in medical area we need the more and more variance, so that not even a single feature is
पीसीए
left.
के बाद वेरिएंस एक एंगल हो गया, लेकिन अगर आपकी डायमेंशनैलिटी रिडक्शन आप 98 फॉर एग्जाम्पल 1145 कह रही हैं, अगर आप 98 करती तो हो सकता है वो 920 रह जाते.
Yes, sir, they were
रह सकते थे
reducing.
ना.
Yes, sir.
और हो सकता है आपको 920 सफिशिएंट थे बिकॉज दे वर आल्सो रिडंडेंट.
But sir,
तो हाउ डू वी नो कि 1145 में रिडंडेंसी खत्म हो गई? व्हाट इज द गारंटी?
sir, with the variance only, like I calculated with 95%, 96%, 97, 98 and 99.
तो वेरिएंस बेस्ड होंगे.
Yes, sir. I considered all the variance, but when I came to know कि as these are the medical related images, we should keep the most variance. We should not keep the variance less. So that's why I selected 99% variance.
ओके.
Okay, so as this discussed in terms of VGG 19, additional layers are there. Now these are the results. In this case, it performed well as compared to the previous one.
स्लाइड पे जाना इससे. ये IEEE ISBI 2019 ML challenge ये क्या है?
Sir, this was one study, and in that study, CNMC dataset was used.
Study means some paper published.
Yes, sir. Paper.
In 2019.
हाँ जी.
और ये मतलब ISBI 2019. What is this?
So this was one challenge. This was one like it was a kind of challenge. It was a kind of competition over there.
अच्छा. Okay.
Based on that, over there, this CNMC problem was discussed, the data set, and then it was evaluated, its results over there, what have been concluded, mentioned.
And this was published in 2019
Yes, sir.
itself?
Yes.
Same year? Okay. ठीक है.
So now the next approach, that is the HP3PGA, 3PGA approach. It is the hybrid parallel 3-parent genetic algorithm, 3-parent genetic algorithm bio-inspired approach. Now mainly it focuses on two things. One is the P3PGA and second one is the 3PGA.
ऐसे ही बता दो.
So first one is the P3PGA and second is the 3PGA. In this case, what we have done, 3-parent genetic algorithm is used over here. So we are generating the initial population and after that applying the genetic algorithm operations onto that.
अच्छा, why 3PGA?
Sir, 3PGA is the advanced variant of 2PGA. We just get the
4PGA कहते हैं तो उससे एडवांस हो जाएगा.
Sir, complexity would also increase as we keep on increasing the parents.
तो कितना कॉम्प्लेक्सिटी में कितना फर्क पड़ जाना था?
Yes, sir. हाँ जी. Because this was the concept we have used 3-parent genetic algorithm. Like there is one mother, but another donor is there, third is the father is there. This is the concept of 3-parent genetic algorithm. Mitochondria nucleus concept is there. Mitochondria is taken from one of the parent, then nucleus is taken from the second parent, and then the crossover is done with the third, and after that the offspring is produced.
Is there 4PGA also medically? Similar approach applied for the female?
Sir, like
Wait, आपने कहा donor और वो from the male side there are 2 parents.
sir, it
Can it be for the female?
No, these are for the like 2 parent from the end of female.
End of female.
Yes,
so
sir.
Now there are maybe 2 parents from the end of male. No idea.
No idea.
Okay.
Okay, so in this P3PGA part was used to find the local best solutions, and to refine that, to find the global best solution, the 3PGA was used. So mainly here when we were using the P3PGA, multiple populations were used over here, and multiple individuals were over here. So then after that that operations of the genetic algorithm was performed continuously to find the best local best solution, and whatever local best solution was found out of the different populations, that solution was provided to the next one, 3PGA. It became the global best solution. Then final search was revolving around that global best solution. So this was one algorithm which was designed. After this algorithm, we have also introduced a neural architecture search. Mainly the purpose of this to create a neural architecture search where automatically the neural architecture is generated by this, and then it is used for the detection of the leukemia. Now, this is the algorithm. So should I explain it like that?
That's it. So normally in presentation you are just supposed to show, speak a crux if you want to tell, otherwise not detailed discussion.
And then this algorithm was firstly designed, then compared with the different 80 benchmarks too. And these were the different transformations, basic bias which were considered, and 10 functions as mentioned in the benchmarking algorithm were used. And what
for your problem, why I should bother about CEC 2021 data? Why not, why I should bother about a CEC 2021 data set? Why should not I focus on your problem specific data set?
Sir, actually firstly actually this is the benchmark suit CEC 2021.
Okay.
It includes these different functions which were complex problem related functions were there, and
My question is still, why should I bother about a CEC data set or those problems? Why should not I directly focus on your data set for applying this architecture and solution?
So I can move to that also.
नहीं, मतलब आपने क्यों किया? मैं आपका कारण पूछ रहा हूँ. Why did you
just to give
do?
the authenticity to my algorithm, like
Okay.
is it worth as compared to others?
It's a good logical answer.
Like here I have compared my algorithm was qualifying out of 80 functions, it was qualifying 49 functions and giving the best performance.
Okay.
So that's why it was used.
अच्छी
Now
बात है.
the neural architecture search, in this neural architecture search, these were the different hyperparameters which are considered: the convolution layers, the number of filters, the size of filter. Their range was initially specified and provided. So this algorithm, with the help of parallel parent genetic algorithm 3 PGA, it was finding the best architecture and keep on moving. So we found the best accuracy in the 82 generation, that is 98.99. So then compared the results with the others here also. So this proposed approach outperformed with 98.99% accuracy for the detection. So to conclude this thesis, and overall in this we have proposed one hybrid soft computing based search and optimization algorithm and 3 soft computing based leukemia detection approaches: VGG 16 PCA PV3C, VGG 19 PCA PV3C, HP3 PGA 3 PGA neural architecture search, and overall HP3 PGA 3 PGA outperformed all the other approaches. Now in future, this model can also be applied to detect the other different types of cancers and diseases for the detection of them. And
When you would like to apply to others, will there be any change into the model or into your algorithm, or only training it over the new data set will be
so
sufficient?
we can train it onto the new data set if we just want to apply the neural architecture search based on the hyperparameters are mentioned in the slide. Like whatever hyperparameters we have selected, if we want to keep the same number of hyperparameters, we need to only change the data set.
Should we keep the same or should we change?
We can change also. So we can change also.
Why should we change?
So it depends, like if we want to change, if we want to keep some another range of the hyperparameters.
Should we do that or not? For a different kind of a cancer or different kind of a disease?
So it's not actually mandatory because our architecture is giving the good results with that specific parameter.
No, but my question is, will the same architecture give results on other type of cancers or other kind of disease detection by simply going for a new data set?
Yes sir, it will give.
If so, that means any solution made by any researcher should be applicable to all other diseases also?
Yes sir, it can be.
If so, that will mean that you designed a solution which has not captured anything specific to leukemia disease prediction. Is it true?
But sir, I checked with this only. I have not checked with the others also.
You checked one is experimental.
Yes sir.
Another is what is your analytical and logical basis of model. Is there anything connecting your architecture with the problem characteristics?
Sir, as per my understanding, like we are providing the images and our approach is providing us the it is providing us the architecture. When it is giving the complete architecture, whatever data set we consider, I think that doesn't make more effect. If we are giving some another data set also, our model is giving the best architecture based on the...
Somebody made for a disease detection of kidney, they made a new architecture where they got. That means that that architecture we can apply for this disease also, and hopefully that may also give good results.
Sir, in case if in that case it is also image based.
Okay, for your case, if I say I apply it to the breast cancer images,
yes, sir.
Can I accept
Sir,
that?
it can accept.
It can accept
Yes,
that. So that makes things so simple that do it for one kind of a disease cancer So doctor's problem will be solved. That now machine learning will find all kinds of cancer diseases by the same solution. So we don't need multiple specialists.
Sir, again it
Although we need multiple cancer specialists as a doctor, but machine learning models don't need multiple specialized machine learning models for different kind of diseases, not only cancer. Ultrasound, I have some stone disease, I have kidney disease, I have a liver disease, I have a lungs disease, I have a heart disease. One machine learning model should be applicable everywhere. So multiple specialized doctors not needed. Once we have an excellent machine learning model, one doctor will be sufficient for all kind of diseases which are image based. Should we believe that?
Sir, actually this mainly model is for the early detection.
So if I target early detection, one model should be good enough for all kind of diseases. Even if you have lungs problem, if you have infection problem, if you have ultrasound issue, because all these will be images.
Yes, sir.
So that will make things for the medical science so easy.
Yes, sir.
Is it possible?
Sir, it's not possible. Still, we need the authenticity of the doctors.
So authenticity of doctors is different, but that means model will be able to do so at least.
Yes, sir.
You feel. चलिए आगे चलें फिर बात करते हैं उसकी भी. Yeah, anything more?
So this model can be applied to find the whenever we want to use certain solutions in the networks also, certain kind of architectures we need to find over there, it can be applied. And most important thing, we can focus the multi-objective. We can in future we can just apply it on the multiple objectives to find the
So what will be multiple objectives in that case?
Sir, multiple in this we are focusing on the accuracy.
So what other objectives will be there?
Other objectives can be the complexities also, computational complexities.
Why should I reduce complexity if I am getting good results? Why should I bother about
Sir,
training complexity? Why should I bother?
So to make the model light.
Acha, okay. So if you reduce the complexity as an objective, can complexity change as an objective function? Can you justify that complexity can be an objective? Complexity, how complexity gets defined? Based on the algorithm you made, based on the architecture you chose. Once it is chosen, during making its optimization, how complexity can change? If the complexity value earlier was, let's say 35, and it says over 23 it is good, but your algorithm complexity is 35, so it will remain 35. How it can change? So how it can be optimized during optimization phase once algorithm complexity is already defined? How it can change?
So like in case if we want to focus on the multiple
I say yes, I want to focus. But how it can be an objective which can be compromised or changed? Objective, it's a fixed value. If you have defined order of n cube complexity or n square log n, it will remain n square log n. Optimization process says let it become n log n. Will the algorithm automatically change because it says not n square log n, n log n? If it says, how will it work? And what will be the meaning of that? So how you can choose it as an objective?
Sir, actually I mean to say, maybe I had not correctly conveyed I mean to say in terms of time. Reduction of time.
Complexity means time itself.
Yes, sir.
Complexity means time complexity, and time complexity equivalently means time execution. And time execution is dependent on algorithm. Once decided, it cannot change. Algorithm change किए बिना time execution time कैसे बदल सकता है? आपका optimization algo तो कहता है कि अगर ये 23 होता तो मेरा ज़्यादा better optimize हो जाता. तो आप अगर 35 मिलीसेकंड लग रहे हैं तो उसको 23 कैसे कर दोगी? जादू कैसे होगा? एल्गोरिथम तो 35 ले ही रहा है ना.
सर एल्गोरिथम में मॉडिफिकेशन करनी पड़ेगी.
तो एल्गोरिथम में मॉडिफिकेशन हुई तो सारा ही बदल गया. फिर तो
आर्किटेक्चर में
तो
उसके हाइपर पैरामीटर्स
तो बदल गया वो तो. लेकिन आपने तो ऑलरेडी वो फ्रीज कर दिया. उसके बाद आप इवोल्यूशनरी एल्गोरिथम तो उसके बाद लगा रही हो ना. ऑप्टिमाइजेशन के लिए लगा रही हो उस टाइम तक. तो बाकी सब तो फ्रीज हो चुका. तो फिर वो कैसे होगा? कैसे हुआ था? तो अब क्या करोगी? इसलिए वो तो छूट गया मल्टी ऑब्जेक्टिव. if you want to talk about multi-objective, what else will be there other than accuracy as an objective function?
हाँ जी.
क्या करोगे?
Sir, not in detail I have studied, but I started about Pareto optimal solutions also.
बट पैरेटो ऑप्टिमल तो तब बनेंगे जब मल्टी ऑब्जेक्टिव बनेगा
Multi-objective
सॉल्यूशन, उसके लिए पहले मल्टीपल ऑब्जेक्टिव डिफाइन करो.
एक्चुअली सर हमारा आइडिया था हो सकता है वो जैसे कार्बन एमिशन मॉडल के टाइम ज़्यादा जनरेट ना हो और जल्द एक्यूरेसी भी आ जाए. कुछ पेपर्स पढ़े थे कि कार्बन एमिशन कम से कम हो, एक्यूरेसी ज़्यादा हो तो कंफ्लिक्टिंग हो जाएगा.
कंफ्लिक्टिंग होने चाहिए, लेकिन इस सिचुएशन में कंफ्लिक्टिंग कौन से हैं? तो मल्टी ऑब्जेक्टिव वर्सेस मल्टी कंस्ट्रेंड सिंगल ऑब्जेक्टिव में फर्क तभी आएगा जब मल्टी ऑब्जेक्टिव में जो आपके कंस्ट्रेंट्स हैं वो कंफ्लिक्टिंग है. अदरवाइज सिंगल ऑब्जेक्टिव और वेटेड कंस्ट्रेंट्स में वर्क करता ही. तो आपके केस में कंस्ट्रेंट्स ऐसे या ऑब्जेक्टिव्स ऐसे कौन से हैं जो कंफ्लिक्टिंग इन नेचर होंगे? एंड ऑब्वियसली कॉम्प्लेक्सिटी क्या है, भी कॉम्प्लेक्सिटी तो एल्गोरिथम की फिक्स हो गई है, वो तो एक तरह से कांस्टेंट हो चुकी है, तब वो तो ऑप्टिमाइज हो ही नहीं सकती. तो और ऑब्जेक्टिव कौन से?
Sir, I'll work on this.
हाँ, क्योंकि फ्यूचर वर्क है तो अभी आपको इसके लिए स्पेयर किया जा सकता है कि दैट यू नीड नॉट आंसर दिस. बट इफ यू क्लेम, इफ यू राइट, you should have clarity that what would you like to do. तो उससे अच्छा कि ना लिखो, क्या जबरदस्ती, अगर नहीं पता है तो लिखना ही है ना?
ठीक है.
और बताओ आगे.
Sir, next is about these are about the publication.
IGIT और इंजीनियरिंग जर्नल्स हैं आपके इसमें. ओके फाइन.
And these are the conference
अच्छा
articles.
तुम ये बताओ कि why didn't you target any SCI जर्नल पब्लिकेशन? आप IGIT इज स्कोपस, दूसरे वाला तो नहीं है, स्कोपस भी नहीं है. चल वाई डिडंट यू टारगेट? ट्राई करती, नहीं होता अलग बात है, क्यों नहीं किया?
कोई किया था सर, अभी थोड़े दिन पहले ही वो
रिजेक्ट हो गया. कोई बात, कहाँ किया?
Evolutionary
कोई स्प्रिंग
evolutionary intelligence.
इवोल्यूशनरी
कोई दिन के बाद वो
रिजेक्ट हो, कोई बात नहीं, ठीक है, बट ट्राई करना चाहिए. और क्योंकि आप फैकल्टी हैं तो यहाँ रहना है तो आपको तो और फोकस करना चाहिए कि अच्छे पब्लिकेशन और भी आएँ. तो ओके, या आगे.
So these were the conferences where I have presented the article.
ओके फाइन.
Sir, it is very small
Copyright?
actually. Yes, sir. Copyright.
फाइन.
This is just the proof of the published
ठीक है.
paper, accepted
ठीक है.
paper.
Okay.
So
So
this is another.
there are some functions which other examiners has also written. Are you aware of these questions?
Yes, sir.
पता है इनका रिप्लाई रिटर्न बनाया है या ओरल बताना चाहोगे?
Sir, written submitted.
रिटर्न सबमिटेड.
Yes, sir.
तो रिटर्न सबमिटेड मेरे पास मेल पे भेजा हुआ है, मुझे ध्यान नहीं है मेरे को. हाँ, मैं इसीलिए कह रहा हूँ, मैंने देखा नहीं है. तो अगर है तो मंगवा लें, तो फिर पूछने की जरूरत नहीं, मैं पढ़ लूँगा इतने. अगर है तो, नहीं है तो मैं एक आधा इनमें से पूछ लेता हूँ. ये नहीं करते वो, ठीक. हाँ, तो फिर तो पूछने पड़ेंगे, फिर तो ये पूछने पड़ेंगे. कई जगह ये भी शेयर किए जा सकते हैं. हाँ, अलग अलग है, मैं इसीलिए हाँ पहले पूछ. हाँ ठीक बात है. हाँ एग्जैक्टली. हाँ इसीलिए कोई वो नहीं है. वही मैं तभी पूछा कि अगर हाँ फिर बोला कि हाँ कहते हैं मैं क्या भी चला दिए. ठीक है. तो बेटा चलिए, वाई वॉज PB3 chosen as an optimization algorithm and how does it compare to other optimization algo like TSO or GSO? Why PB3C instead of others optimization algorithm? Primarily the question is this.
PB3C, sir, this is one. Initially, I had BBBC, then PB3C from here, parallel Big Bang Big Crunch. This is the optimization because it is based on the Big Bang Big Crunch theory. So I had gone through this. I had gone through the other also like
PSO, KCO, GSA
also. So explore.
उनमें से क्यों ये?
Sir, I found this better and
in terms of results, experimental results,
and its methodology also. It is expanding. Then it is coming to the global best as per my requirement.
वो तो हर एल्गोरिथम कहता है कि ये हमारे वाला बेस्ट है, ग्लोबल बेस्ट पे जल्दी पहुंचता है, हमारे वाले में एक्सप्लोरेशन ज़्यादा है, हमारे वाले में एक्सप्लॉयटेशन बढ़िया है, धीरे धीरे कन्वर्ज जल्दी होता है। सारे एल्गोरिथम यही क्लेम करते हैं। तो आपकी जस्टिफिकेशन क्या है? क्योंकि वो तो एल्गोरिथम वाले हर कोई लिखता है, हर एक अपने अपने पेपर में लिखते हैं और तभी सारे छपे भी हुए हैं। तो आपका आंसर क्या है? नहीं पता। अच्छा,
ये ना बेंचमार्क किया था मैंने।
ओके,
एक्चुअली मेरा ही पेपर था PhD।
अच्छा,
जो पुरानी एल्गोरिथम्स थी उनसे बेंचमार्क फंक्शन
से निकाला।
तो मैंने सीधे कहा था कि ये
Okay.
तो आउट परफॉर्म कर रहा है।
ओके, हाउ डू यू एड्रेस पोटेंशियल चैलेंजेस रिलेटेड टू डाटा सेट बायस और ओवरफिटिंग? वॉज दिस अ केस? आपके ओवरफिटिंग या कोई इश्यूज थे या नहीं थे? अगर हो तो कैसे एड्रेस करोगी? ओवरफिटिंग मालूम है?
Like overfitting, where I have considered when the size of the data set was small, then over there were the chances of the overfitting. Here in our case, the size of the data set is already large and it's sufficient. So I did not see anything related to overfitting.
अच्छा, why was VGG16 chosen over new architectures like ResNet, DenseNet or EfficientNet?
So
Why not those? Why VGG16?
VGG16 is when I analyzed the various transfer learning models like VGG16, VGG19, ResNet 50 and all. So then VGG16 was one of the model which was giving the good results, plus it was not very much complex also. Here the number of layers are very more. In case of ResNet 90, ResNet 50 convolution layers were being used for the results, whereas in this case, so I started with the light weighted model.
Okay. Is it possible to deploy the proposed model in real-time clinical settings, or any constraints will be there in terms of computation?
Sir, it can be applied.
Any constraints you feel?
If the data set is given to us in the same term, like images are there, we have done the image-based classification over here, so we can take the images from the blood smear and then this model can be applied over there.
Acha. Okay, there are few more questions. What are the alternates to PCA for dimensionality reduction? Is it necessary to consider all dimensions after applying PCA? Is there any data loss issue?
So we are having LDA. LDA can be applied. TSNEs, then other dimensionality
Okay.
reductions, they can be.
So did you explore and find why LDA versus PCA any performance issues?
When I had explored these things, PCA versus all these, so I came to know when it comes to the classification in terms of the images and all, so it gives good results, PCA.
How did you come to know?
Sir, I have just studied, gone through the
From where?
from these resources, from
कोई रिसोर्स याद है जो ये कहता है?
नहीं, मतलब जहाँ से पढ़ा था कि
हाँ, मतलब
PCA
कहाँ से पढ़ा था? मतलब अगर चैट जीपीटी से पढ़ा था वो अलग बात
नहीं
होगी।
सर, चैट जीपीटी से नहीं।
हाँ, फिर कहाँ से पढ़ा? तो कौन से रेफरेंसेस कहते हैं ये बात?
मतलब डिफरेंट इनके मेथडोलॉजी, लाइक दे आर PCA इज यूज्ड फॉर सम पैटर्न्स फॉर सर्टेन इमेजेस ओवर देयर. वी कैन यूज SNE also, this linear discriminant analysis also, but as in our case we had to introduce the non-linearity and the data was non-linear, so we had used PCA.
Can generative AI play a role in this problem? Generative AI का थोड़ा आइडिया तो होगा?
हाँ जी, हाँ जी, हाँ
Is it useful for your problem or not?
It can, so no.
अगर करेंगे तो हाँ, नहीं है तो कोई बात नहीं, नथिंग रॉन्ग। मतलब ऐसा नहीं है कि सारे क्वेश्चंस का आंसर आना ही चाहिए, नथिंग रॉन्ग। ओके, द प्रॉब्लम हैज बीन सॉल्व्ड यूजिंग PB3C, BBC and GA. Now, the most important part, can you correlate your problem characteristics with their equivalent physical? Like if you say PB3C, शुरू की पॉपुलेशन में क्या जनरेट होगा?
शुरू की पॉपुलेशन में सर फीचर्स, कैंडिडेट सॉल्यूशंस,
तो जो कैंडिडेट सॉल्यूशन है वो वर्सेस आपकी जो फीचर्स थे, क्या सारे फीचर्स को एक वेक्टर मान के मैं उसको एक कैंडिडेट सॉल्यूशन मान लूँ? फिजिकली कैन दैट बी इंटरप्रेटेड लाइक दिस?
यस सर, इट कैन बी.
अच्छा, अब आप बताओ, आपने क्रॉसओवर लगा दिया,
यस सर.
इसके फीचर्स और दूसरे किसी और डिफरेंट डायरेक्शन के पिक्सल के फीचर्स क्रॉसओवर से तो कंबाइन हो गए।
So then we need to focus on this kind of crossover only.
लेकिन
like in the same column.
आपने तो 3GA क्रॉसओवर लगाया, 3 पेरेंट वाला, तो उसमें तो हो गए।
But
और
point.
वो पॉइंट नॉन एग्जिस्टिंग पॉइंट बन गया।
फीचर्स नहीं है सर इसमें आर्किटेक्चर।
नहीं, ये कह रही है यार पिक्चर्स है, मैंने तभी तो पूछा कि नाउ फिजिकली इंटरप्रेट।
But in case of sir PB3C वाला मैं कह रही हूँ, VGG16 VGG19 में feature.
अगर आप क्रॉसओवर लगाएंगे तो क्रॉसओवर में कहीं के पिक्सल्स की वैल्यूज किसी और पॉइंट या किसी और लैटिट्यूड लैटिट्यूड के पिक्सल से कंबाइन हो गई, तो हो सकता है वो तो नॉन एग्जिस्टिंग कोई चीज़ थी और आपने उसके बेस पे मॉडल को ट्रेन किया।
But sir, in case of like point to point crossover, then can't it be
अच्छा,
the
पॉइंट
same?
टू पॉइंट में भी बताओ, एक पॉइंट अलग लोकेशन का था, दूसरा पॉइंट कोई बिल्कुल अलग लोकेशन का, उस डिजीज वाले एरिया से ही नहीं है, उस शेप में ही नहीं एग्जिस्ट करता, दोनों को मिक्स करके एक नया एरिया या नया टेक्सचर बन गया हो, तो जो एग्जिस्ट ही नहीं कर रहा था मेरी इमेज में, तो फिर वो कैसे वर्क कर जाएगा। और आपने एक्सप्लोरेशन के लिए क्रॉसओवर लगाओगी।
Yes sir.
तो फिर ये तो बिल्कुल ही अलग डायरेक्शन में पॉपुलेशन को ले गए और उसने ट्रेन कर दिया, तो कैसे चलेगा? समझे? मतलब वाई इज ऑलमोस्ट ओवर, लेकिन ये क्वेश्चन लिखा था तो मैं इसीलिए उससे पूछ रहा हूँ। तो इस तरह से बहुत सारी चीज़ें हो गई। एनीवे, चलिए या तो व्हाट नेक्स्ट? क्वेश्चन आर ओवर। एनी क्वेश्चन फ्रॉम द ऑडियंस? एनीवन फ्रॉम द ऑडियंस साइड?
Sir,
Yeah, supervisor can also ask if it's any questions. Others, audience also can ask. Then I'll conclude after this.
on a lighter note, what was the major challenge throughout your research you faced, and how did you try to overcome that? Any experience? So you took a lot of time in doing all these things, right?
Yes sir.
So what was the major challenge or complication that came on your way, and how did you overcome it?
So challenge was at every phase, like to decide the methodology, to decide the algorithm, which one has to be selected, then to learn the new technologies.
The most challenging part.
Most challenging thing is in terms of the resources for the execution, when we are doing the execution. So sometimes like on 3-4 laptops, on 3-4 systems I had executed the thing. So every time I was, most of the times I was getting the message the kernel has died in my system also. So when it is executed for 7 to 8 hours, after that I was getting that kind of message and it was completely shut. So this was the most challenging thing which I faced.
Hardware issues.
Yes.
They are required.
Yeah. Any other query from the audience? Any query? So, okay, fine. Then I'll conclude. So, Navpreet, well done. Nicely presented. You were well prepared, confident. Your presentation was also nicely planned. Time was also nicely planned, except a few glitches which I have already discussed. Fine, but overall, you worked hard. That was visible. I suppose that you have rehearsed also with supervisor and maybe alone. And how many rehearsals did you do? Count.
So rehearsals for this presentation.
For this presentation. Mock presentation. Done या नहीं?
Did you do it at home?
घर पे या
Sir, only 1 or 2.
Okay. So 1 or 2.
That was
That
not
means
complete.
I would like to tell you, sir, she has been awarded as one of the best teachers in
Okay.
this school.
Very
Of a good level.
nice.
And she has won a medal for that also.
Acha.
That is why I was
Ah.
observing one thing, and when she was presenting, and suddenly वो measurement marker उठा के लिखने लगा।
हाँ, वो टीचर वाली आदत होती है।
वो है।
That's so. तो
टेक्निक
वही हाँ
बट वी डिस्कस
techniques तो good मतलब with only one presentation effectively you did very well. Ideally it must be rehearsed even if you are a good teacher.
Yes.
It's an exam and before exam full mock presentation rehearsal at least 2 is must. and that will give you much more confidence. But because you are a good teacher, so you know that art, so you could present it well. That's good anyway. Secondly, the work done is also quite good. Quality is also good. The volume of the work is also good. I have evaluated many theses of LPU, taken Viva also. So found every time there are very few where little eyebrows are raised over the kind of a work done or the direction chosen. Results coming doesn't mean a good quality work. Good publication also doesn't mean good quality work now. There are people who publish that in the summer it can rain like this and it can become winter and it can get published into a transactions paper. That in June 2029 there will be a winter like January and they will prove it and it will be published. But that obviously everybody will say it will be nonsense, so it can get published. So there is nowadays that kind of a research also being done, but your research has a logical connections and that. Having said that, so overall relatively you did good work. Knowing the LPU or other private universities standard, the PhD work is worth as a PhD, no doubt. Of course, higher levels are always desirable. Coming to the other side, where you should realize that lot more was to be done also from an ideal perspective. Relatively, you are better than many others. But if you say a good PhD doesn't mean only experimentally assuming that this model did good results, hence this model to be used. You used PCA because PCA gave good results. You used PB3 because PB3C gives results. You used this architecture, it started giving good results. You tried with for hit and trial method of different parameters. Wherever results came, you are happy. That's an average kind of a research work. Research doesn't start like this. Research has to start by finding first what architecture will be suitable to my problem and why, how problem characteristics get mapped via this, what kind of a filters, what kind of a features, what kind of the pre-processing, what kind of a reduction is relevant to my domain, how that can be achieved from which kind of a models, and then attempting some of those applicables only, and then going for experimentation results is the ideal way of conducting a research, which is ideal, which you have not done. Similarly, when you say that your model will be applicable to, say, lung disease detection or any other kind of a disease or image-based ones, that becomes a tall claim. And not only tall claim, logically it has no basis. If that is true, that means there is a universal solution possible for all kind of such problems, which is impossible. It cannot happen. And if it is so, then there is no need to do thousands of research papers publication every year for every problem. Then whatever you are doing, that should be applicable for crop disease dissection also. That should be applicable for the cultivation also. That should be applicable in the armed forces also. क्योंकि हमने तो कुछ किया ही नहीं प्रॉब्लम स्पेसिफिक. हम तो कह रहे हैं हमारा बढ़िया आर्किटेक्चर बना, इमेजेस पे ट्रेन होता है. अब मुझे पाकिस्तान पे अटैक करना है तो मुझे यही वाला मॉडल उठाऊं, वहाँ की इमेज लूंगा, वहाँ ट्रेन करूंगा 50 इमेजेस पे, उसके बाद वो वहीं से बता दिया करेगा कि उग्रवादी कहाँ बैठा है. माई मॉडल शुड वर्क. भाई यहाँ से वो मेरे को कैंसर बता रहा है, वहाँ वो बता देगा. ट्रेन होने की देर है, तो सिर्फ ट्रेनिंग पर डिपेंडेंट है. मॉडल तो बढ़िया ही. If it is so, that means it is. And if my statement is correct, then your model must have a mapping with the problem, which is missing. We use those things as a black box and not knowing why this model for my problem. So that is the weakest link of the research. Ideally, that is where every researcher, not only you or for future, you should focus. ठीक है? So, but yes, having said that, well done. Congratulations, you are a good worker, hard worker, logically well done. Congratulations.
Thank
So
you, sir.
I can announce you as a Dr. Navpreet now. Formally, just Preet ji will announce through his process. So congratulations to Dr. Navpreet for good work.
Thank you so
And
much.
congratulations to the supervisor, to the department, and all others, and to LPU for another good team.
Thank you.
Thank you so much.
Congratulations.
Thank you, sir. Thank you.